just for this week, I'm gonna let you take these lab pages home. Don't leave them with me because I want you to take them to study for your midterm next week, but bring them next week so you can leave them with me and I can check them off. Okay, so for today's lab, DNA isolation and electrophoresis, that's what we're gonna do. We're gonna start instead of 3 o'clock starting, I'm gonna start at 3:30 next week. Again, if you get here early, it's just the door will be locked, that way I can turn everything on, and that way you don't have to wait for 10 minutes while I do all that. And we'll just say 3:30. You sit out there and need that time to study. Alright, so we are going to be on page 69. Okay, we're gonna do some DNA isolation. Alright, what has DNA on this planet? Yeah, all this living stuff. So if you're considered a living cell, you have to have some DNA, whether you're a plant, bacteria, all that good stuff. Okay, so we're actually gonna pull the DNA out of some kiwi today. Kiwi, right? And it's kind of like a kiwi smoothie kind of going on. You would do something similar if we were gonna do like paternity testing. So forensics, criminal testing, anything like that, you can isolate the DNA out. Now here's the little model of DNA. Remember everybody, it's a double helix, you got A, T, G, C's. It's one of those macromolecules. So that macromolecule lab a few weeks ago, we did food. And I told you we weren't gonna test DNA even though it's a macromolecule. Now we're gonna do DNA lab. Do you eat your food to get DNA out of it?
No.
No. Sure. Right? You can transform. That's a GMO, right? So that's why we didn't really cover it in that lab. The procedures are completely different. So for like Benedict's, Biurettes, all that stuff last time, those were the other macromolecules. So we're going to concentrate on DNA today. Okay, so for this helix, we kind of go over in lecture. Some of you have lecture, some of you don't. Kind of the parts and pieces of it. So when we do forensics, we're figuring out how yours line up. So those bases in there, those are those A, T, G, C's. Right? And so everybody has a different combination of these letters. So you have roughly 3 billion of these letters in your cells. Okay, they control all of this stuff. This is your blueprint. So that makes you unique, but a lot of it's gonna be the same between us. So in forensics, because I used to work for law enforcement, I used to do this. Okay, what do we get in the lab? Oh yeah, where does it come from? Oh, we get all kinds of bodily fluids because all cells have it in there. So when you do that, you pull the DNA out and you're looking for the arrangement of these letters. Okay, we look at a small piece that's unique between all of us. So every one of us in this room are 99.9% alike with these letters, but that small percentage out of 3 billion is a lot. That's what puts you in jail, right? That's right? Yeah, when I say it like that, you remember it, right? It's not fancy for criminal forensic, but it means go to jail, right? That's forensics. You also use it for baby daddy. Right, paternity. Do we ever check mom? No. Sometimes you have to. Yeah, sometimes they steal babies, they kidnap them. We've had cases where there was actually a big group of kids kidnapped, and this was in a whole different country. They found them, thought they found them 10, 15 years later. How do you match that? With mom or dad, we did this for like World Trade Center. After disasters, you don't have to have a lot. DNA is microscopic. But think about this. We have 3 billion of those letters crammed into your nucleus. That's a lot. Okay, that's a lot of these letters. So that's kind of what we're looking at today. So we've got to get the DNA out of our kiwi first. And then we're going to do one of those gel electrophoresis. So we're going to take these letters, kind of simulated, we're going to run it through like a battery. DNA is negative, so we're going to separate out those bands. And I'll kind of show you what it would look like for a crime scene. We're just going to kind of simulate it though with kiwi and some food color. Make sense?
You said it's negative? Is that what you're saying?
Yeah. And so when we get through and insert the battery, I'll kind of go through that too. But all of this is based on this structure. These A, T, G, C's and the chemistry of it. All right, so we're going to start on page 69. This is explaining all that background stuff. Right, so again, what does your DNA do? Right, it's your blueprint, so it's everything, but it's still just a combination of these letters. So your normal introduction, then on page 71, here's the procedure. Okay, there's a lot of steps to this. I'm gonna kind of show you where all this is at, then I'm gonna kind of turn you loose a little bit. You know, last few times I kind of walked you through. This time you're gonna do it on your own. We're at midterm. You should know how to measure, weigh, read the temperatures, all that good stuff now. But I'm gonna kind of show you where all this is at. Okay. So up here's all your materials again. You've got weigh boats, all this good stuff. Most of this is in here. Okay, I do have a note, and just make a note when you're reading yours. And it says for the inoculating tube, instead of a glass stirring rod in the materials and methods, there's a metal tool in here somewhere. We'll dig in here and find one. You got one? Yeah.
So no glass rods?
Yeah, and there may be a glass rod in yours, but there's also like this silver handle thing with a little hook on there. So kind of either or, depending on, either one's in there. The other stuff, the food coloring, all that, that's gonna come in a few minutes. But your beakers, all that stuff you're gonna find in there. All right, so for the kiwi, right, you guys can read all these directions. You're gonna weigh out 30 grams of this kiwi smoothie. Anybody like kiwi?
Yes.
Why not? Well, it looks good, but does it taste? How does it taste?
After you cut off the
Okay. So you know when we do this too, it's gonna say you need cold alcohol. Okay, cold ethanol. It's back here, it's on ice. Don't get it too soon. Okay, if you do, it's gonna warm up, but it's always back here on ice. I'm not gonna get it too yet. Smoothie's in the fridge, and it looks like a smoothie. We take kiwi and just grind it up. What color is kiwi? Green. It's gonna look like a green kind of smoothie. So here's your ground up kiwi. Got all the seeds. Ooh, yummy, huh? You're gonna do this by table. Oh yeah, they feel it. They kind of grind it all. Just like it. Well, you know how kiwis got the little. Yeah. And so there's a spoon back here for you. So it's 30 grams. So you should have a big wafer in there. Oh, it says they use a 40 ml beaker. It's gonna tell you, no, not that one. It's gonna tell you to use a big wave boat. Up here, they said you should have a 400 ml beaker. May I have a, that one's small. So it should be this size. Yeah, it's bigger than what you think it would be. You do the same thing that you would do with the wave boat. You put it on there, make sure you zero it. Okay, 30 grams is gonna be a nice chunk. Remember last week when we did yeast and all that? How much was a gram of powder? Yeah, so you're gonna do 30 grams this time. So it's gonna be a nice chunk of kiwi. Okay. So as you go through though, it's gonna talk about funnels and some coffee filters, all that stuff should be in here. Okay, so I'm gonna let you guys get set up and start weighing the kiwi, and then I'm gonna kind of walk you through the steps, but it's gonna take a few minutes for everybody to kind of bottleneck right there to the kiwi. All right, so you need 30 grams, so send somebody from your group. Okay, there's a spoon back there. It's already in some liquid, so once you get your 30 grams, it says pour that 30 grams in your beaker. It'll already be in there. It says use 30 ml, your graduated cylinder, and 60, so it says pour, and so read this next part. Okay, it's got some soap and all that in it. Huh? No, you're gonna use the beaker. Oh, and we didn't get to skip number 2 actually.
Did you say skip it?
Yeah, because they already have it in the solution. They've already mixed it, so you just need the 30 grams. I got another note up here. Yeah, have somebody weigh your 30 grams over here. Somebody will come measure out 30 mLs. You gotta mix them, and then we're gonna go to the next step. That way we can divide and conquer. It's in Canvas. That way if something's wrong, people don't see it, they can use it. But you can literally you do it. There is in Canvas, because I have to hand out some video, they don't. Yeah, and you can pull out another jar if you need to. There's 3 jars over there if you want to kind of. When you're done with your graduated cylinder, make sure you rinse it out. 30.
The homogenation solution, she's already got that.
Now, when you get that, kind of swirl it and mix it, and it's gotta go in this 60-degree water bath over here. Okay, when it goes in there, write down the time, because we're gonna have to wait 15 minutes. You don't wait the whole 15 minutes, it won't work. That's the stuff. Yep, and then you just put 30 mL. And the whole beaker after you kind of swirl it. No, it's all mixed in one. Yeah, so then, oh, well, the finals are coming later.
Or spot.
So,
We're putting homogenation solutions in 60-degree Celsius water bath.
now, once you get these mixed, you do have a glass stirring rod in there. Stir that soapy stuff up with your kiwi. Okay, it's gonna have to go in this water bath over here. It's also at 60 degrees, but write down the time or set a timer. It's gotta sit in there 15 minutes, but every 5 minutes you have to stop and stir it. Okay, if not, it won't mix it well enough to get the DNA out. You just have somebody every 5 minutes.
I think it's black, sir. What time?
That's what time it's gonna actually precipitate out the DNA. And then make sure you rinse your stuff.
Oh, you're just doing the time.
Now, when you mix them together, stir them up really well, otherwise this won't work very well. Okay.
And so every 5
You
minutes,
just stir it up, and then somebody from your group every 5 minutes go stir it again really well. Now, in this salt solution, there's a couple of things. The detergent, the soap breaks your membranes, okay, and the salt is gonna help us get the DNA out. Okay, so you're gonna have soapy salt basically. Oh, yeah, besides, yeah. Is it floating, James, or are you good? Okay, if you need to, you can sit a weight on top of it again, but it's not floating. And make sure you just kind of stir it. Okay, now, you've got kiwi in there, that's gonna have DNA in every cell. In the homogenation solution, you have got a soap, that's to break membrane. Okay, the other thing is the salt to kind of precipitate out that DNA. It's gotta sit in there 15 minutes. Yeah, so stir it really good. Stick it in the second incubator for 15 minutes, but have somebody in your group every 5 minutes go stir it pretty good again. You're good.
and you start at 3:14.
Okay, so while this is sitting, somebody else in your group needs to go to step 5. You're gonna set up a filter system. Okay, so if you use your graduated cylinder and or funnel, make sure it is rinsed. Okay, there's a coffee filter in your bucket in the center. gonna use this as a filter. So the coffee filter, the funnel, and then your 25 ml graduated cylinder. Okay, so we're gonna end up filtering our mush here in a minute. So you want a 25 ml.
It's exactly 25.
Okay, so just make sure you have a 25 ml. Now, after your 15 minutes are up, okay, we're gonna pull that beaker out of hot water over there to 60 degrees, and it's gonna go into that ice bath, okay, for about 5 minutes. You gotta time it, but this time stir it every 2 minutes.
Yep.
Yep. Yeah. And you just gotta watch it so a bunch of water doesn't get down in there. Okay, otherwise, you gotta start. You can get the water in there. Okay, if you don't have your lab reports, there's a stack of them up here, so while everybody's dividing conquering, you're gonna want these. Your grades are in Canvas. I didn't put all of the grades on here so that everybody didn't see grades. But it's in Canvas. I just want you to have all of these so you can study this next week for your midterm. Okay, so make sure you pick these up.
Forgot some of these issues were different. And it, from 5:30 and from 6:06.
Yeah, you can go start it.
Did you find yours?
Yeah, I found
Okay.
one. There's another one here. I
Yeah, it
wanna make sure
depends
there's
on
no
the
more.
order.
It's all good. No big
Yeah,
deal.
make sure it's in there because it's not in there. I think
It's all good. I'm not missing any
Okay, then
grades,
you're good.
so I should be okay one way or another.
yeah, I just always think to check. Every once in a while I get some that I don't have any
Yeah, no big deal.
names on.
No big deal. We'll make it happen.
Yeah, there's one here. So yeah.
Quick question, did you happen to give her the grade for this one, or did you give me the grade? Because this is mine, but she made a copy.
I'm sure I gave it to her.
Oh,
Send me an
man.
email. Send me an
Okay.
email.
Okay.
Because I probably was like, why does she have 2 grades? But it looked like a copy, so I didn't think anything of it. So yeah, you're going to have to send me an email.
Okay.
Okay, everybody's in the water bath. Just don't forget to stir every couple of minutes. Everybody done with the smoothie? I'll stick it back in the fridge. When you get done with there and you put it on the coal, make sure somebody else stirs it, but you don't get water all in your smoothie mixture. Well, with DNA, you have to time these pretty well, because if you short time it or do it too long, this won't work. So you make sure you watch that time.
So what are we doing now?
Okay, I'm gonna flip to this while we're waiting. You see this chart up here? I want to talk a little bit about how chemistry works with your DNA. So you know how you see how it's a double helix? Okay, if you folded this double helix out, it would look like a ladder, right? If you could imagine that. Looks like this. Okay, your ladder, kind of the part where you would put your hands on your ladder, that is a sugar, right? This is the deoxy part. So when we say DNA, that's deoxyribose, that's the sugar. Okay. You also have to have a phosphate. So this is just a phosphate group. Okay, so it's a sugar. Phosphate would be the part we put our hands. The rungs of your ladder where you put your feet, that's going to be those bases, the ATGCs. So on this side, and I'm just going to do ATGC. Right? Looks something like this. Okay, so that makes up the rung. How DNA works, there's only 4 letters. Okay, they will only hook together in a certain combination. So when you look at the chemistry, A always binds with T, G always binds with C. There's one of these questions on your midterm next week. So make sure you know this. Okay, so here, if A is on this side, guess what's on the opposite side of the DNA? T. It's like a negative, negative. You can't put 2 negatives together. 2 A's won't go together. An A and a G won't go together. So this side is going to be a T, an A, a C, and a G. And on your midterm, I'm going to give you this long piece of DNA. Not long, it's like 5 or 6 letters. See? And it looks something like this. I'm going to say, what would the other half of the DNA look like? All you have to do is match the A's with the T's, the G's with the C's. Everybody following that? And if you come up here and look, you'll see blue, pink, blue, pink. That would be the AT, the yellow, green would be the GC. If you don't hook them together like that, it doesn't come together this way. In biology, if it's not the right structure, it will not function correctly. That's your blueprint, right? Your code for everything. If it doesn't look like this, you don't function, okay? Depending on what's mismatched. That makes sense? So for that table, after your little discussion questions, there's like 4 or 5, there's a table right here. That's what you're matching up. So if A is here, T would go here. C would go with G. That's what they're asking you to do. Okay. Now, while we're doing that, how much, we've got 5-10 minutes, don't we? Okay, so when you look at this, let's answer some of these questions. Why do you think the kiwi was mashed into a smoothie?
To pull out the DNA.
Yeah.
Because the liquid is coming.
Yeah, so imagine this. What do you have to leave behind at a crime scene? How much?
Just a drop.
Just a drop, not even enough you can see. You know, they'll go in and find it. You gotta kind of break it up. It's hard for us to get the DNA out of something really thick and solid. So we kind of have to mash it up so we can get the DNA out of all the cells. Okay. Now, when you look at this, why do you hypothesize? What would, again, you guys can answer that. It's your hypothesis. Just pick something, and again, you can kind of figure that out as we go. Now, why did we heat it? And again, that's going to talk about that in your introduction. It helps kind of break all this up. It's going to get rid of some of those enzymes. Okay, that's going to break up your DNA. So this is written in the introduction piece too. It'll tell you specifically heat is used to destroy enzymes that can break down DNA. Now for, what was the, is DNA soluble in ethanol? We're going to get to this in a few minutes. After we cool it, we're going to add some ethanol, right? And it precipitates out. So is DNA going to dissolve in ethanol? Right, and you're going to see this. It creates one of those white layers. So that's what it's asking about. Okay, and it'll tell you that in the intro. The next piece is just to fill out that DNA. Okay, after this, we're going to run it and do an agarose gel electrophoresis. When you see electro, what does that mean? Electricity. And so on your tables too, you'll see these things with red and black cords. You ever jumped a car battery off? What's the red and the black mean?
Negative.
Positive, negative, or ground. We're going to do the same thing, but we're going to run it through our DNA. Remember diffusion? Remember my tall person versus me? Right, I'm little, I'm like 5'2" on a good day. Right, remember James back there? He's like a foot taller than I am. Well, I don't, you guys are all taller than me. Right? Anybody shorter than 5'2"? Oh, see? See? There we go. Right? Everybody's taller than me. Who's gonna get through the door faster? I am, right? I'm shorter, I don't have to duck. Okay, remember diffusion? Which one went faster? The little one. Guess what the little pieces of DNA are gonna do? Go faster, especially when we basically electrocute them, right? We're gonna put a little charge through them. Right, but if DNA is negative, which way you gotta go? Toward a positive, right? So we just have to hook this up. We're gonna use food coloring so you can actually see this, because usually it's clear on clear. And we're gonna see this kind of go through the little battery and all that. And so we just gotta wait till all this other stuff's done, and then we're gonna set that up. Okay? Those are gonna run for 30 minutes when we get the battery hooked up. I'm gonna talk about the midterm then. Very good? Okay. So how much time do we have for the water baths? Okay, don't forget to be stirring them. Okay, when you pull them out of that hot bath, the 60-degree, make sure you follow this. If you're gonna end up putting it on the cold, okay, 2 minutes. Have somebody.
Forwarding that for 5 minutes.
Yeah, it's like 2 minutes, or what is it, 5 minutes? 5 minutes, stir it every 2. Okay, so just have somebody stand back there and watch it and stir it.
Step one.
So as soon as we're done here, we put it in.
Yeah. Can you? No, it's not that.
Used to test for DNA or is it strain?
And then we're gonna filter it. You only need like 5 mL of that filtrate though to put into that next step. Don't play with it.
Strands. Yes. Oh, no, I was just a question in my mind, because I was thinking about this with the banana on the nuclear, and I thought they said that they used the consistency of the banana to test for DNA.
Oh,
I just had
when
a
we
flip.
pull that DNA up in a few minutes, when we do the alcohol and all that precipitate, it's like snotty. That's what they're, you know how you bake
So
the
that's-
banana, it's like, that's what the food gonna be.
And so that's where the DNA would be in the liquid DNA.
Yeah, it's the stringy stuff.
I got you.
And that's what they meant. And it's, I would say it's snottier than a banana, but you know what I mean? That's, but yeah, it's not gonna be flaky or anything like that. It's gonna really in stringy. Your front or my front?
Yeah.
Oh, here's your stir rod if you need it.
That's subjective, I choose my finger.
Yeah, so just kiwi and some soap and alcohol, some salt. Ice bath, 5 minutes, stir it every 2. Okay, then you come back to your table and you're gonna filter it through your coffee filter. You're gonna collect at least 5 mL. We're gonna need 5 mL to go into this large test tube on your desk to do this next step to actually get the DNA out. Now, if you've used your graduated cylinders, all that, make sure you've rinsed those before you measure the alcohol or anything like that with them. Otherwise, it kind of messes all this up. DNA is super sensitive. Because what happens in forensic when I sneeze on my criminal sample? My DNA goes on it. What's going to happen then? Oh, well, usually you weren't accused. It throws the case out though. Because that was a technical error. See, and yeah, and for me, my DNA, because I've worked for law enforcement, is in that FBI database. So, you know, once you're arrested, they do all this stuff for you. You know, they do the cheek swab or the blood. They put those letters in a database. The whole world has access in law enforcement to that. So it doesn't matter where you go, it follows you.
So you work for the FBI?
I work for the Florida Department of Law Enforcement, for the state FBI. Yep. In a lab, and you guys, it's never that fast. You know, on TV, it's like, poof, you did it.
Yeah.
No, the cool thing about DNA though is getting people out of jail. Now that we have technology, you know, they go back and look at that and it's like, no way y'all could have done it. And some some of those people are on death row for like 30, 40 years. Yeah, it's not that hot. Yeah. And they do it a little fancier than this in the criminal life. But it's basically all the same thing. It's alcohol and some salt and soap. I've heard of people doing that. I saw something this past week where people found their siblings committing brandy and
I
stuff.
told him I was gonna do it, he's like, I go, he's like, I had a wild past, he's like, you're not doing that.
I found out I had a sibling in the hospital.
I thought that he was serious.
I mean, they knew about it the whole time, and in fact, my dad had one, but then a couple of years before that, we found out my mom had one too, and both of my brothers were 6 days apart from different parents. It was insane.
I don't know, I had two half siblings when I was 16. So he's like,
What's
I know you
the
got two, he's like, we find it out.
Oh, that's what comes out. So once you put in your coffee filter, what collects in your flat or in your graduated cylinder is the filtrate. You need at least 5 ml in there. Again, just be careful back there with the ice water so you don't get water in it. Have one of your, group members stand back there and stir it every 2 minutes. It's not too bad, but you know how they'll tilt in the water sometimes you don't want to get ice or water in there. And then you're gonna come back and filter it. 5 ml and it goes in your big test tube. Yep. Yeah, once you're done with your filter part, all that gets tossed and rinsed and all that good stuff. Put back on your table.
Dr. Sonia, what's that word that?
Oh, homogeneous.
Yes, the oil and the water, don't come together, but if,
Oh, hydrophilic, hydrophobic?
yeah,
Yeah.
hydrophobic.
Hydrophobic.
Yeah,
That's what you're trying to bust up. That's why we use soap and detergent.
But that's
It's
what I
to
was-
break up the grease, the
Right.
lipid, the oil.
right.
Yeah, and so we're trying to do that with the cells. That's what.
I remember you talking to me.
Oh, it's okay.
Just can't remember what the word was.
One of those fancy science words. That's all I say. What does that mean in English? Right back here? And again, the ethanol's back there. Just make sure it's still cold. Oh, yeah, you know where it's at.
Alright.
Yeah, so the filter, and it's gonna get kind of thick, it takes a couple minutes, and then you're gonna put it in a big test tube. Yeah, if that let it drip long enough, see where you get at least 5 ml out of there. Yeah, so make sure you have plenty, and then all that just gets rinsed. Now, if the alcohol comes up to that, when it's I know, and you only need 5 mLs of it.
These are, we have 2 of them. And this one is only 25 ml.
Yeah, it's gonna take a couple minutes for the filter to, you know, when you're done, this will go in your actually need 5 mLs to go in here, and then your alcohol. And when it says to try to get the DNA, use this instead of a stir bottle. Because that stir bottle will make it wrong. And then that'll go in the trash, and then everything just gets rinsed.
Stir it close to the bottom.
That's me. Big yes.
And this is the sanitation, like just the 5 mLs, that'll go in your test tube, and slowly do the alcohol, this thing is that white later. Yep, this whole thing will go in the trash, and you can wash it, clean all this stuff, too. Regular trash.
Yeah, do you have an extra cover filter? They can look like the whole
Oh, yeah, here's one. I was gonna say, where'd it go?
I don't even stir it, just a little closer to the bottom.
Oh, and it poked it. Yeah, just gotta refilter it.
Yep.
Right. I was like, we don't have to start over with it being
Usually it works this time.
Does anybody have 2 funnels?
You have 2 funnels. Nope.
So you and I are complete opposite.
So the other one is being.
Is it smooth or?
Yeah.